The overall goal of this method is to automate the analysis of a Caenorhabditis elegans germline to study its nuclei, protein, and cytoskeletal distribution.
This method can help answer the key questions in the C. elegans field about the number of nuclei and sperm, nuclei distribution, and cytoskeletal structure of the germline. The main advantages of this technique are: it reduces the analysis time, increases the sample size, and eliminates the human error associated with the manual analysis.
To image the samples, place slides with stained germlines into the slide holder above the 63x objective of a confocal microscope, and locate a germline. Focus on the top of the germline and mark it with the confocal microscope software. When the total germline thickness has been established, define the thickness of each slice up to 0.5 micrometers and mark the complete germline. Then, start the acquisition, scanning each slice 8 times and averaging the values to improve the image quality. When all of the images have been obtained, import the images to an appropriate image analysis software program, and use the Surface Function tool to select the mitotic region. An image of the distal end of the germline will appear. Cancel the automatic surface creation wizard and manually draw a region of interest around the mitotic.